our noses above water. You're doing okay.
Yeah, exactly.
I think we can get started. It's 4 o'clock. I'm sure there'll be more people joining in the next few minutes. So hi everyone, my name's Adam Shellard. I'm one of the organizers of the cell migration seminars. The rest are on camera. You can see Jen, you can see Becky, and Ashrith is around somewhere. There he is. So the idea is we're going to have a seminar once a week at this time for 1 hour. The format might change week to week, so sometimes it might be 1 speaker or 2 speakers, maybe 3 speakers, but we'll see how it goes. Thank you all for registering. We've had 900 people register from all over the world. You can see in the bottom left, there's that heat map across the world where everyone's registered. If you have any questions or feedback, you can contact us the Twitter and email or on the screen. If you have any technical problems, we probably won't be able to help because none of us are IT people, but Becky will try her best. So you can try and message, if you're on Zoom, you can message moderator_Becky or the same on YouTube, just write in the chat. And you'll be getting an email from us every week with the information for the seminars. So we aim to send that once a week. As a brief preview, next week we've got Robert Insel from University of Glasgow, and the week after that we've got Sally Holm-Badagowicz from the University of Chicago. But that will all be information you'll see in the email. The capacity on Zoom, as I mentioned in the email, is 300. So if you can't access on Zoom, just go onto the YouTube link. And then a final 2 things before I introduce the speaker. The first is once the speakers finish, we can have a go at everyone unmuting their microphones and giving a real round of applause rather than a virtual one, if you want to try that. But then obviously, please mute yourself after that. And if you have any questions for the speaker, if you're watching on YouTube Live, there's a chat window where you can type your question, and then I'll ask it on your behalf. If you're on Zoom, there's the same chat window. You have 2 options. Either you type your question and I'll ask it on your behalf, or if you want to ask the question yourself, write the word question or the letter Q into the chat, and then I'll call your name, and then you can unmute yourself. And if you want to as well, put your camera on, then you can ask the question and then just mute yourself and turn your camera off again. Okay, I think that's all the admin done. So today's speaker, thank you, Jeff Fredberg, who is a professor of bioengineering and physiology at Harvard University. Jeff's lab has been one of the leaders in studying material and physical properties of morphogenesis and cell migration, which in the past used to be more single cells, but now it's more collectives. And he's going to be talking today about the geometry of cell jamming. So thanks, Jeff. I'm going to get rid of my screen if you want to share yours.
Okay. Your screen. Okay. So everyone can see that? Good. Well, I'm super excited to be doing this, and I want to thank the organizers for inviting me. It's a great honor, and this is a new medium for all of us. So this is terrific, and that 900 people registered is phenomenal. So it's, I guess, fringe benefit of or silver lining of COVID. So I'm going to talk about geometry of cell jamming and the unjamming transition. So one of the things, it didn't really lead us here, but one of the things we started to think about was there are certain tensions in epithelial biology. That is to say, it's shown on the right actually is an artist's rendition of the airway epithelium, which is the system we work in most. But there's times when this epithelium has to be solid-like, where all the cells have to sit in place. But there's other times when the cells have to migrate and become fluid-like, like in wound healing or growth. There's also a tension between, on the one hand, precision. These cells have to do certain immune functions and barrier functions, but they also have to have a certain amount of plasticity to change in response to various kinds of insults. There's also a question of kind of order versus disorder. A lot of people in the literature talk about an epithelium as being kind of a hexagonal grid, but actually it's a very disordered grid. It's not really hexagonal at all. And then finally, I think I wouldn't surprise any of the people who are listening to this talk to say that epithelium is a dynamic tissue. But at the same time, with all those dynamics, there's an energetic consequence. It has to be economical in terms of its energy utilization. There's not an unlimited energy budget. So all these factors are in tension with one another. And I think that the idea of jamming and unjamming helps to shed a kind of a different kind of a light on these tensions. In the background of everything I'm going to say is the question of disorder in biology. So we all know that kind of biology is messy compared to physics or engineering. And the question I want to kind of deal with subliminally is messiness just noise? Is it just biological noise, or is it actually necessary for biological functions? So those are the things I want to talk about. So first, who cares about unjamming anyways? So what I'm going to talk about is epithelial cell unjamming. I'm going to do it in the context of asthma, development, and tumor dynamics. So these are all unfinished stories, and I'm going to do these in those 3 acts. And the protagonist is epithelial cell unjamming. So first, asthma. So this is bronchial epithelium. You can see ciliated cells, goblet cells, other cells. In the airway, this epithelium is a pseudostratified epithelium. It has many cell types, only some of which are shown here: serous cells, ciliated cells, goblet cells, basal cells. We were interested in this epithelium in the context of asthma. So a key hallmark of asthma is remodeling of the airway in a variety of different ways. And we know that the epithelium is involved in this, as is viral infection, which are both deeply implicated. So this story starts about 8 years ago, where I went to this gentleman. Some of you might recognize him. It's Jeffrey Drazen. He has just stepped down as being chairman of the editor-in-chief of the New England Journal of Medicine. And he and I have been collaborators for many, many years. And he had a lab right next to mine at Harvard School of Public Health. And I went to him 8 years ago and I said, Jeff, we're starting to study collective cellular migration. And you've been studying these epithelial cells in airways for decades, actually. And we have reason to think that there may be an unusual migratory phenotype of these cells in asthma. So when you look in the microscope, what do you see in terms of epithelial cell migration? And he said to me, Jeff, he said, nobody studies that. These cells they just sit there. They're sedentary. They sit there like tiles covering a floor. And as the barrier becomes degraded, it's like grout that fails in the floor and it becomes leaky. He said, yeah, there are some people who studied wound scratch healing assays, but for the most part, no one's interested in migration of airway epithelial cells. So I went to his trainee at the time, Jinar Park, who's now an assistant professor in our program. I said to Jinar, what do you know about migration of these cells? And she said, well, to her knowledge, she said, no one's ever measured it. So why don't we do that? So we did. So these are human bronchial epithelial cells. These are primary cells. They're measured in air-liquid interface culture. They become well differentiated and pseudostratified. So this is thought to be the best in vitro model of airway epithelium. So I'm going to play the movie. First, the one on the left is the control. And if you don't see much moving, that's because it isn't moving much. You can see a little jiggling going on. On the right are precisely the same kind of cells, prepared in the same way, except they're exposed to a compressive stress, which we think is involved in bronchospasm. I'll come back to that later. And when you compress these cells with an apical to basal compression in the physiologic range, you see this, which is really a striking migratory phenotype, everything else being the same. And you can also notice interesting changes of cell shape compared to the left. So we were really interested in understanding this better. But what I want you to appreciate, and I'm going to show you, is that these cellular motions in the left versus the right, they're not only differences in the amount of motion, and the differences in amount are huge, they're orders of magnitude, but there's a difference in the quality of the motion. It's qualitatively different, differences in kind. And it turns out that difference is captured actually in a theoretical model that was developed by Max B. who's at Northeastern University now. He did this with Lisa Manning originally in Syracuse. And he developed a model which we found very helpful. And here's the model shown on the left, control and compressed cells. And so here on the left is his representation of control cells, non-asthmatic cells. And you can see that they're jiggling, but they don't actually go anywhere. And it's as if each cell is kind of trapped in an energy well. It's just like a marble in the bottom of a cereal bowl. It can vibrate, but it can't actually go anywhere. So there's an energy barrier. Each cell is kind of caged by its neighbors because of the crowding. The matrix as a whole is solid-like. That is to say, if a cell jiggles from its position, it always snaps back to its original position. So it's solid-like or elastic. And we call that situation jammed. Now on the right is, with only changing a few things, you get a different kind of a phenotype. These cells, each cell, if you track one long enough, each cell will eventually sample everywhere in the space, as if it's diffusive. And it's as if the energy barrier went away. And in fact, the theory, which I'll tell you more about, actually says the energy barrier goes to zero in this case. So there's no energy barrier that's impeding these cells. They can go anywhere. And it's as if they're uncaged and the situation is fluid-like. And we call this unjammed. So if you were to track the center of each cell, you see on the left that each cell is kind of restricted to a given position, caged by its neighbors. The cells are stuck. They're as if they're frozen. We call that jammed. And on the right, you can see the trajectories go all over the place. The cells become unstuck. And we say this is if the matrix as a whole is melted, and we call that unjammed. Now, the theory is important. Not that I believe the theory. I don't really believe any theory, but they're useful guides for thought. They point you in directions and they generate hypotheses. So what this theory said, among other parameters that we'll get into, it says that there's a key balance between tension and adhesion along the cell-cell junction. So as you all know, along the cell-cell junction, there are actomyosin motors that are disposed. And when those are activated, they make a tension that tends to make the cell-cell junction shorter. They're contractile after all. On the other hand, there are adhesion molecules. There's a whole slew of different kinds of adhesion molecules. But for example, E-cadherin, homotypic binding, that the minimum energy configuration of that situation is where the cell-cell junction is long. That adhesion wants to make cell-cell junctions longer. So these two forces, or energies, if you will, are in opposition to one another. Tension making the cell-cell junction shorter, adhesion making it longer. And the theory says when tensional forces dominate over adhesive forces, you get a situation like on the left. Cells become jammed. But on the other hand, when adhesive energies become more important than tensile energies, the energy barrier goes to zero, and the theory says the system becomes unjammed. Okay, so before I go further, I just want to give you a potted history of the unjamming transition. Now, this is neither complete nor just, but it's kind of how I view the field. I'm going to start at the end. Recently, actually, Jen Mitchell, one of our hosts today, has a paper it's posted on the archive. It's in review presently, and she shows definitively that at least in this system, the unjamming transition has nothing to do with the epithelial to mesenchymal transition. Those are two totally different things. So all of you probably know about EMT, but the UJT is a different beast altogether. I'm not going to talk about that, but you can look it up on the archive. So, oops, there we go. Okay, so the first mention, to my knowledge, of cell jamming in the literature was a paper from my lab where we were measuring traction forces by the monolayer on the substrate. This is work led by Javi Tripak, who's now in Barcelona. And in it, we looked at these force distributions, and they had strange fat-tailed distributions, which were things similar to what had been seen in inanimate systems that are jammed, like gravel, granular systems. And so we suggested the idea in that paper that jamming might be going on amongst cells, but a different kind of jamming than in granular matter. A few years later, Donna J. Tombe looked at the intercellular forces, the forces actually transmitted from cell to cell in the cellular collective, and found further evidence that there may be jamming. Then Tommy Angelini, who's now in Florida, who was then working with David Waits and with us, he published a really important paper in PNAS showing that the dynamics of the cellular motions were not actually diffusive at all. They're actually something called, physicists call glassy dynamics, which I'm not going to get into, but that's a hallmark of jam systems. It gave another hint about jamming. And then again, Xavi Tripat in Barcelona published this paper where he showed that there are waves that occur in these layers, and they might be waves of unjamming. But it was actually this paper by Jinnah Park in our lab that really nailed down the fact that epithelial layers can jam and unjam using a variety of sophisticated metrics that I'm not going to go into, but came from the physics literature. We showed that these systems really are jammed. They show all the hallmarks of jamming. So this was actually a key paper. And then it was followed shortly thereafter by work from Joseph Kassov's group in Leipzig, where they looked at organoid plasticity and argued that those systems were jammed as well. Now, all of these together are in vitro systems. So they suggest unjamming in vitro systems, but it still left the possibility that this is all just an artifact of culture. Then this paper came more recently, 2018, by Lior Adia, who was working in my lab and now is at Ben Gurion University in Israel. And amongst the many kinds of data in that paper, we looked at gastrulation in Drosophila in the living embryo. I'm going to show you some of these data later, and showed that unjamming occurs in that living system. That was followed shortly thereafter by Oche Campas at UC Berkeley, where he looked at vertebrate axis elongation in the zebrafish and argued that jamming is a key factor there. And then Celeste Nelson at Princeton looked at embryonic avian airway branching and found evidence of unjamming. And this paper in the imaginal disc wound of the Drosophila also found evidence of jamming. And these altogether are in vivo systems. And this told us that cell jamming and unjamming, it's a real thing. It's a real biological thing. It's not just an artifact of culture. And then I'm going to move my own picture out of the way here so I can see it. Then in the cancer literature, jamming started to catch on as well with these important papers that appeared. So unjamming, the idea is that in a tumor, cells will migrate more if they're unjammed. And I'll show you some data about that later. Then I think I have to tip my hat also to the theoreticians, because it turns out it was the conceptual background that really supported, it was the foundation of all this work. The first and maybe most important paper was from Frank Huliker, which looked at cell packing and fluidization in some theoretical studies. They didn't connect this to jamming, but what they modeled was jamming and unjamming. Christina Marchetti and her group looked at active jamming. Max B, I've already mentioned, looked at density independent transition. So it's not just about crowding, it's about other things. And they looked at motility driven unjamming as well. And then Nir Gov looked at active cell jamming. And then also I want to mention at the lower left, Salvatore Toccato at Princeton looked at the mathematics of jam matter. Papers I really don't understand very well. They're very theoretical, but I think there's a lot there for people who can understand it. So all this led us to a hypothesis or question. The hypothesis was that maybe the default homeostatic state of epithelium is quiescent, solid-like, and jammed. On the other hand, in asthma, it seems as if someone yelled fire. Did someone mute whoever that was? Okay, sounded like a young person. It's as if someone yelled fire in a crowded theater, and the cells become agitated. There is no fire, there is no wound, there are no inflammatory mediators, yet the cells are on high alert nonetheless. They start agitating, they become fluid-like and go through rearrangements. And we thought maybe this is a maladaptive repair phenotype. There is no wound, but the cells are looking for a wound to heal, but there is no wound to heal. And they start laying down matrix and doing other things we don't like. Now, you may be thinking a good critical argument is that you're just using jamming as a synonym for migrating. How do you know that jamming has anything to do with this? Well, we think that there's a way. So if jamming mechanism were false, how could you really know? So there is a way, and it's because this same theory from Lisa Manning and Max B lead to I find to be a remarkable mathematical prediction. And it's just so simple, it's incredible. What their theory says is that if, for example, on the left is solid-like and on the right here is fluid-like, they said the transition from solid-like to fluid-like is marked by a simple but critical change of cell shape. It's just cell shape. Shape marks this transition. And lots of things could be going on, but you'll know that if it's jammed or unjammed by shape. And you can see it with your own eyes here from the model. And the critical number, it turns out there's a variety of metrics of cell shape, but the two we've used, one is the perimeter of the cell divided by the square root of area. So all you need is a sophisticated tool of a ruler, and you take your ruler and you measure these things. You measure the perimeter and you measure the area, and the perimeter divided by the square root of area, we call it Q. That's a shape factor. And the theory says you go from fluid to solid when the shape factor is 3.813. It's just a number. It's like pi, 3.14, but this is 3.81. Or you can use the aspect ratio, which is the length to the width ratio, and it's 1.18. Now, the interesting thing is this is a pure number. There are plenty of parameters in the model, but in terms of this shape index, they don't matter. The transition from fluid to solid always happens at 3.813. It's an absolute number, and there's no curve fitting. It doesn't make any difference what parameters you choose. This is a geometric index. So we wondered, how does this work? So we turned to the data. And so these are studies from Park. And so here on the left, you see the measured shape index Q. And you can see that jamming threshold 3.813 is shown there with the arrow on the lower left. And here it's some familiar objects. You can see what their shape index is: circle, hexagon, pentagon. Pentagon is darn close to the critical number 3.81. Square, triangle. So that will give you an idea of the range of these numbers. Now, we knew that when we studied these human bronchial epithelial cells in culture, they take some time to mature. And until they're mature, they migrate like mad. But by the time you get to day 8, the migration stops, and the hypothesis is that they were jammed. So when we looked at the shape index, this is what we found. Just as the cells come to day 8 and stop migrating, it's precisely then that their shape index goes to 3.81 with a high degree of statistical confidence. We were astounded by this concordance. When we looked at cells from asthmatics, we know that, this is a change in the time scale. Cells from asthmatics mature more slowly. They migrate a lot. And we know even by day 14, they're tending towards jamming, but they're not quite jammed. And when we looked at the shape index of those cells, again, they become close to 3.81, but not quite there. So again, this is a concordance. They're not quite stopped migrating, and they're not quite jammed by the geometric criteria. So this was important because it told us that cell shape gives us a structural signature of underlying migratory dynamics. It gave us a hold on that. So just to summarize what I've told you so far, we have jammed system on the left, unjammed system on the right. With maturation, cells tend to progress towards jamming. And with asthma, there's a tendency to go the other way. And we thought maybe this is representing some kind of a dysmotor phenotype. So these were the data at that time. And, in the lab, happy. We were really happy about this. This was a remarkable concordance between physics and biology that I haven't seen very often. But the more I looked at these data, the more I became troubled by them. There was something about this that bothered me. And eventually, I figured out what that was. If you look at the change in this mean shape index, the changes are actually small. They're small compared with the actual variability of the data, which is actually pretty big. And I said, oh, so how meaningful is this? Well, we went back to the biostatisticians and say, look, biology is messy. The statistics are good. Take yes for an answer. But that really didn't satisfy me in terms of understanding what was going on here. I was wondering, what are these? What is this variability? Is it measurement error? Is it observer variability? Actually, these were measured by hand by Jen Mitchell, tracing them by hand, laborious work. Or is it real biological disorder? And that's when Lior Adia came into the lab. And he said, well, let's look at that. He said, look, your measurements so far from the Park paper were all phase contrast. We can use fluorescent imaging to get much better handle on the cell-cell junctions. You trace those by hand. We can use auto-segmentation algorithms. You looked at a few donors. We can look at more human donors. And you looked at hundreds of cells. We can look at hundreds of thousands of cells. So let's do that. And so he did. And our null hypothesis was that cell shape was the critical factor, and the variability was just biological noise. And we didn't really have to pay attention to the biological noise. What he found was that these distributions turned out to be very well defined. So here are those same data. And now on the right, here are distributions for other data. On the top panel, human bronchial epithelial cells, but also MDCK cells, living Drosophila, and the computational model on the bottom. And all these distributions can be collapsed. This is shown versus aspect ratio. You can collapse them if you plot them aspect ratio -1 over the mean aspect ratio -1. They're very well behaved. Those are the histograms for the distribution shown at the left. And they follow something called a K gamma distribution, where the formula is shown here, that has only one free parameter, which is K. And what that means is, if you look at where it says computational model, the mean and the standard deviation always have to go together in a one family distribution like this. So in a K gamma distribution, the standard deviation, the variability of the shape goes with the mean shape. Those can't get disconnected if this is the proper distribution. So I'm going to come back in a bit to talk about what this means in terms of mechanism. But before I do that, we asked the question, how general is this result? We just check the time. I think we're doing okay. And this brings me to development. So about the same time, Yasha Shamra came into the lab as a postdoc. And she was very critical of everything we were doing. It's good. It's good when a postdoc or a student is critical. I like that. And she said, look, everything you've done is in vitro. Everything you've done is in flat cell culture. And you've only looked at one cell type, human bronchial epithelial cells. She said, in vivo veritas. Why don't you look at some real living system? Spread your wings. And so I said, okay, fine. I don't know how to do that. What do you suggest? And she said, how about this, Drosophila? So we decided to actually look at gastrulation, the Drosophila embryo, and look at mesoderm and endoderm that come from a uniform blastula. So here's the embryo of Drosophila, and it's shown one of the early events is the formation of the ventral furrow, which is shown here. And that was something that had been studied in great detail by Eric Wieschaus, who won the Nobel Prize for this work. And see, these are some of his beautiful images showing this ventral furrow formation. And we said, oh, this is actually a terrific model because it's a living system, but there are changes of cell shape, they rearrange and they migrate. So we could look at this. And in terms of asthmatic airway epithelium, which was our main interest, it was kind of, does this have anything to do with that? Or maybe does it have everything to do with it? So we wanted to look at this in greater detail. So here's some fluorescent imaging. And I have to point out, we were helped in all this by Adam Martin at MIT, who's a developmental biologist who uses the Drosophila model all the time. And so here's a movie. This is 15 minutes compressed into 15 seconds. And you can see the formation of the ventral furrow there. So our hypothesis was that to move, cells must unjam, as manifest in the cell geometry. So here, I'm going to play for you a movie in a second. And so here is a graph on the right showing cell shape as an aspect ratio and shape variation on the y-axis, which is how variable cell shapes are. The gray data represent the data from our human bronchial epithelial cells. Those were from the Park paper, showing that as the cells matured, you go down and to the left along this relationship. And that solid line is not a fit to the data. That's actually from the B. Manning theory with no adjustable parameters. So that's an absolute prediction from the theory. And you can see it goes along with those data from human bronchial epithelial cells pretty well. The solid black dot in the lower left means absolute jamming in an amorphous system. And the triangle there represents what we saw in the Drosophila embryo before gastrulation started. And we were surprised and pleased to see it fell right on that line. Now I'm going to play the movie showing what happens with gastrulation. On the lower left, by the way, color coded for aspect ratio. And as you can see, as the Drosophila unjams, it goes right up along the same relationship as did the human bronchial epithelial cells. Now those were maturing, they were going down and to the left, but this system is going up and to the right, but along the same relationship. So to move, cells must unjam, we think, as manifest in cell geometry. And we looked at a variety of mutants in this system, and it held up in all those cases. So now the left-hand axis here is shape variation. That's about disorder, but how variable are cell shapes? How noisy is it? How messy is it? And the question is, do you need that variability? In other words, it goes together one-to-one with cell shape. The more the cells are elongated, higher aspect ratio, the more the shape variation. They go together one-to-one. But do you really need that shape variation to get migration? And that kind of reminded me of this quote from the literature, the artistic literature. A certain type of perfection can be realized can only be realized through a limitless accumulation of the imperfect. That is to say, you need this variability to make the system work. So what's the nature of this disorder? So on the next slide, I'm going to talk about a little bit of physics. So if you're not into physics, just bear with me for one slide. And it comes from granular matter. We're soft matter physicists have been thinking about this for a long time. And in particular, kind of a big deal in soft matter physics is what's called the Edwards conjecture, which was originally suggested by Edwards in 1989. And it has to do with systems like granular systems like sand in a sand pail, gravel, emulsions, slurries, things like that. So think about sand in a sand pail at the beach. That system is actually jammed. You can make it flow by tipping the pail over, but it's jammed. And the question is, how does a system like this work? The particles can move under certain circumstances, but thermal agitation is not big enough to move a sand grain. Sand grains are too big. So this is the Edwards conjecture. He said in a disordered athermal system, so this is athermal, thermal forces aren't big enough to move these particles around. He suggested that all jammed packings are equally likely. So if you calculate the number of packings, the number of different ways you could pack sand into a pail, it's like more than the number of electrons in the universe. It's like a huge, huge number of different kinds of configurations that you can do. And what that means is the distributional entropy is maximized. Now we know that that's true in certain equilibrium systems, but this is an non-equilibrium system where the particles are stuck and can't move. So in thermal systems, this is the formal definition of temperature. He suggested that in athermal systems, volume, that is the free volume around a particle, plays the place that energy does in a thermodynamic system. And then you can define, if you believe that, then you can define an effective temperature that relates volume changes to entropy. That gives you an effective temperature. And it turns out that this was proven, this hypothesis, and we found that just as volume variation, it turns out it predicts a K-gamma distribution, just as we found in cells. And shape variation follows this same kind of distribution in cellular systems. And the reason for saying this is it leads to a very simple relationship between an effective temperature and either the variation of the of the cell shapes or the mean cell shapes. Those tied together, and you can see that here. Here's the same kind of graph, shape variation versus shape. And you can see that this T effective says if you go down and to the left, that system is jammed, cold, frozen, and stuck. So that has an effective temperature that's low. And on the right, you get a system that's hot, melted, and migratory. Higher cell shapes, higher shape ratios, aspect ratios, and more shape variability. So let me get on to tumor dynamics. I think we're still okay on time. So this is what I'm going to tell you is actually, it's just been posted on the bioRxiv. It's in review. This is work from Wendy Kang in my lab and Jacobo Ferruzzi, who's working with Mohammed Zaman at BU. And we looked at cancer cell lines, MDA, MB231. These are an invasive cancer cell line. And Jacobo knew how to make spheroids out of these cells. And we examined the physics or the biophysics of these systems at 3 different scales of length. So this is showing this tumor-like spheroid embedded in collagen, rat tail collagen, at a particular concentration, which I'll tell you about. And you can see it makes invasive tongues that penetrate into the collagen. We were struck by something that actually a lot of people sort of know, but no one had really focused on. And we found that quantitative but small changes in the ECM density beget qualitative and switch-like changes in the invasion modality. So what do I mean by that? I'll show you. So here, if you reduce the collagen concentration from 4 milligrams per milliliter to 2 milligrams per milliliter, you go from, on the right, invasive tongues, like rivers of cells that are flowing into the matrix. Instead, you shed individual cells and cell clusters that get shed from the tumor, not these invasive tongues. So what we found was, and I'm going to show you data for this, that the core of this multicellular tumor is solid-like and jammed. The periphery is liquid-like and unjammed. And so the implication is that there's some kind of melting, an equivalent of melting going on. Whereas here, again, we get a solid-like jammed core, but we get a gas-like cell scattering. Cell scattering is not new, but we're putting it into a new interpretation, a new context. And that suggests sublimation. So sublimation is the process that when it snows, and even if the temperature stays below freezing, the snow can disappear. It goes from the solid state of crystal water into gas without ever going through a liquid-like phase. So this to us looked like sublimation. And third, we found, and I'll show you these data, that compared with the cells in the core, the cells at the invasive front at the periphery turned out to be bigger, longer, softer, faster, and stronger, which a lot of this came as a surprise to us. So I'll show you these data. So another kind of question was, if you look at a different cell type, a less invasive or non-invasive cell type like an FCF, MCF10A, you see on the lower left, on the lower matrix density, you see a little bit of invasive tongue, but at the higher matrix density, you don't see anything, and you don't see any gas-like behavior. So what's going on here? So we looked at this more closely, and these were data generated by Jacobo. He looked at the behavior at 4 different collagen densities, relatively, closely spaced, 1, 2, 3, and 4 milligrams per milliliter. And you can see the images there. And below those, you can see the rendering, the confocal rendering of cells that are either contiguous within the cluster, which are shown in blue, or cells that are disconnected and are migrating in a gas-like fashion into the matrix. And Jacobo was able to quantify this behavior, and that's shown in the graph on the right. And you can see early, when you first put these cells into the matrix, at day 0, you don't see much of anything, and then things evolve in time. But by the time you get to day 3, the distribution, this is actually showing the number of single cells that you find in the collagen matrix as a function of collagen concentration. It evolves to what, to my eye, you could look at this as like a dose-response relationship, but to me, it almost looks like a switch. I mean, in biology, that's about as close as you can get to a switch. We didn't do finer gradations to see if it was how sharp this was, but it looked like you go from one behavior at about 2.5 milligrams per milliliter, you go to a different behavior. And that's where Z Chen came into the picture. Z Chen is on the faculty at Dartmouth, and he does a variety of things, including modeling. So he looked at these data and he developed a computational model, a little different than what Max P had done. He looks at the cells and he puts in adhesion and stiffness and propulsion for individual cells in the cluster, and he puts them in this is a two-dimensional model, and he puts them into a matrix, which is represented by pillars or posts that the cells have to migrate through or around. And what he found was this. If you look here, I don't know if you can see my pointer, but in the liquid-like case, you see here, you get invasive tongues that are starting to invade into the matrix. On the left-hand axis is collagen density, and I showed you data for different collagen densities. If you lower the collagen density, you come down here where it says gas-like, and instead of getting invasive tongues, you get a gas-like escape of cells and cell clusters. And then this solid line kind of separates gas-like behavior from liquid-like behavior. On the x-axis is cell motility. And in this model, it's like propulsion, random propulsion forces. So if you go here from liquid-like, if you lower the propulsive forces, you go over to the left and you get solid-like behavior. So everything over here in the left is solid-like in this space. Everywhere in this wedge-shaped region here is liquid-like, and everything in the lower right is gas-like. And these solid lines represent the boundaries between these different kinds of behaviors. So these are like coexistence lines. And you can even find a boundary between gas-like behavior and solid-like behavior. That's sublimation. And there, that's a triple point where you can get all three phases at once. So this theory, this model, computational model, suggests a coexistence phase diagram. That is to say, it's a new physical picture of tumor cell invasion as non-equilibrium phase separation. So if there are any engineers in the audience, if you've ever looked at a phase diagram for water, ice, liquid, vapor, it looks kind of like this, but that's an equilibrium, thermodynamic equilibrium system. These are non-equilibrium systems, but it looks like a non-equilibrium phase separation, maybe a way to think about tumor invasion. Cellular forces. Let me look at, I'm running short of time. I think I may skip this. Let me just show you this movie. This is actually showing invasive behavior of these are MCF10A cells in a matrix. This is from Ming Guo. And it's interesting, you can see in the center of the tumor, cells aren't moving much, but you can see out in the periphery, cells are actually trafficking back and forth. So looking like it's jammed here and not jammed here. And I just want to make a long story short. This is the method that Ming used. This was actually published in Cell back in 2014. He could actually measure the fluctuating forces inside of a cell. And in the interest of time, I'm going to actually skip over the method and just show you actually the data. So this is the mean square displacement of those cells, either in a peripheral branch, the periphery of the tumor, or the core of the tumor. And you can see that in the core, there's not much motion. The mean square displacements are small. In the periphery, they're greater. And out in the branch, they're even greater still. This is a log scale. So that's quite a big change, about 10-fold. He was able to look at the force fluctuations inside the cell. You've got to just stick your finger, shrink yourself down, stick your finger inside a cell and try to feel what the force fluctuations are like. Thermal fluctuations are way the heck down here. Now, this is a log scale, 6 orders of magnitude. This is the force fluctuation spectrum inside the cell. And this is the blue is near the core. The orange is in the periphery, is in the periphery. And this is way out in the branches. That's a 10-fold difference here. And if you deplete ATP, you drop by 2 to 3 orders of magnitude. So that's the force fluctuation spectrum. And what he found was that the Migration velocities are smallest in the core and biggest out in the periphery. Move this out of the way. Cytoplastic stiffness, this is something I don't understand. If anyone has an insight, I'd be interested to hear it. The cells in the center are stiff, but those out in the periphery are much less stiff by about a factor of 5. And if you look at nuclear volumes, they vary quite a lot too. The cells in the center are compressed, if you will. They're smaller, and out in the periphery, the nuclear volumes and cellular volumes as well are bigger. So cells actually have large-scale intermittent fluxes that take cells from the periphery to the center and back and forth. And as they do that, the cells are getting bigger, longer, softer, faster, and stronger. And these are the data that support that. And it's all about the location, because it's not about an individual cell. The cell changes its properties as it traffics back and forth to the periphery. And it turns out, if you look at not MCF10A cells, but actually human breast cancer explants, you find much the same thing. So what I've shown you so far is jamming versus unjamming, maturation, development, asthma, mechanotransduction, and I haven't shown you these data, but it turns out as well, they all unjam the cell, the default state and the excited state. Let me conclude by just saying a few words about energy metabolism. So these are new data. They're not yet published, but we're working on a manuscript right now. And it's Steve DeCamp, who came into the lab 3 years ago and wanted to look at metabolism. And I told him, Steve, I don't know anything about metabolism. We don't have any tools to measure metabolism. But he was insistent, and he went ahead and measured metabolism. And it's a good thing that he did. So we took a page from the book of Javi Tripat, the Sarah Picamel paper. We made a PDMS mask, and we coated cells. We plated cells in the middle on a polyacrylamide gel, and then you lift the mask, which creates free space into which the cells can migrate. And you can see here at the top, this is from Picamel, Sarah Picamel. When you lift the barrier, the cells start to extend into the free space, flatten, and migrate. And these are data, again, from Sarah Picamel, showing phase contrast images, early, mid-time, and late time, showing the layer spread. This is looking at the velocity from particle image velocimetry, showing the velocities arise first at the edges. This is showing the traction forces, which again arise first at the edges and then spread into the layer. And this is actually the stresses within the layer transmitted from cell to cell. So all of that is measurable, and we measured it. What Steve did was he superposed on this metabolism in a variety of ways. First, he used a paradox bioprobe to get the NAD to NADH ratio, which is the redox potential. And so here's what he found. So this is the aspect ratio of the cell. This is before you lift the barrier. The left is the aspect ratio of the cell spreading area, their speed, traction forces, and the redox ratio. And if you look at 4 hours later and 24 hours later, you can see that the barrier has, the cell front has advanced. The aspect ratios have gotten extended. Cells have become elongated. Cells have become spread. They've become faster. The traction forces have grown. And interestingly, the redox potential has fallen. It goes down. He also looked at glucose uptake, shown here. And if you look at the right, this is the glucose uptake per cell. And at the leading edge, that goes way up. And you can see the fluorescent images on the left. And he also looked finally at the mitochondrial membrane potential. And again, if you can see here, these are some controls. But here, again, the mitochondrial membrane potential goes up at the leading edge. So what this told us, none of this is surprising, but it documented the following, that the redox potential, glucose uptake, and MMP all suggest that the jam phase is energetically economical, but that the unjam phase is energetically expensive. Again, that's not surprising, but it's nice to see that documented, and you'll see why in a second. So this is kind of the summary slide. In addition to being the default state, the jam state being solid-like, non-migratory, frozen, it's also economical, and it's, if you will, with an effective temperature, it's cold. But the excited state is unjammed. It's fluid-like, migratory, it's unstuck and melted, and it's actually expensive. And we can call that kind of a hot state. And so this, I'm going to conclude with kind of where this leaves us with the unjamming hypothesis. These are the things we're thinking about now. First, the cell jamming, I would argue, is the evolutionary adaptation of the epithelial collective to the pressure of maintaining homeostatic barrier functions at minimum metabolic cost. I think that's pretty important. Let me say it again. Cell jamming is the evolutionary adaptation of the epithelial collective to the pressure of maintaining homeostatic barrier function at minimum metabolic cost. If true, that would be pretty important. Number two, in a manner that is energetically costly, but unlike the EMT, does not compromise barrier function. Of course, when you have EMT, you lose effective barrier function. So in a manner that is energetically costly, but is not like EMT, it does not compromise barrier function, cell unjamming and the unjamming transition provide for a necessary fluid-like phase that allows for episodes of epithelial migration and resulting mechanical plasticity as required for tissue development, remodeling, and wound repair. Again, I think that's pretty important. That is to say, unjamming, you can have plasticity, but you don't have to compromise barrier function. Jen Mitchell showed that in her paper that's in bioRxiv in review, at least in that one system. And finally, unjamming can become hijacked in disease like asthma and cancer. So I would argue in closing that the unjamming transition is fundamental to epithelial biology. It's not an epiphenomenon. This is fundamental to epithelial biology. And I would argue further that if as much or if not more so than EMT, you need to be thinking about cell jamming and the unjamming transition. So I just want to give a plug. On YouTube, I've got 2 talks. One is about how to write. So if you're struggling with how to write or you want to try to improve your writing, this is everything I know about how to improve your writing. And there's a second one is about scientific speaking. If you didn't like what I had to say today, you don't want to look at the second one. But if you like it at all, you might want to look at this. It's everything I know about how to give a good talk. So the acknowledgements, these are the most important people. Of course, I believe that you're only as good as your trainees. And these are trainees and collaborators. Chan Young Park runs my lab. Jennifer Mitchell has done a lot of the pioneering work for the jamming transition. Yasha Sharma, I've mentioned. Jay Kim did a lot of cancer studies. Jim Butler is our theoretician, applied physicist. I've mentioned Lior Adia. Wendy Kame and Jinar Park, crucial members of our team. Jacoby Farouzi and Mohammed Zaman at BU. Ming Guo, I wasn't able to describe his methods, but he's a fantastic experimentalist at MIT. And his trainee, Yulong Han. Adam Martin is the Drosophila biologist. Zi Chen and Catalina at Dartmouth did those modeling studies, and at Northeastern, Max B and Herb Levine, and those are the funding sources. So I'll stop there and take any questions that you have. Thanks for listening.
Thank you very much, Jeff. If everyone wants to unmute and give an applause, they can do that.
Thank you.
Thanks, everyone. You can mute yourselves again now. Okay, so just a reminder, if you have questions on YouTube, there's a 30-second delay there. So just ask it in the chat. And if you have questions on Zoom, you can either ask it in the chat, post your question and I'll say it, or you can type the word question or the letter Q and I'll call on you. So Roberto is with the first question.
Yeah. Hello, I'm Roberto Mayo from UCL.
Yeah, hi Roberto. We've met once. Yes.
Yeah, yes, hi. So first, great talk. But also would like to congratulate the organizer for organizing this great activity. So I have two questions, and one is a very technical, I think a technical question. I mean, it's about the Drosophila data that you show. If I understand correctly, what you analyzed was the changing the cell shape in a 2D situation.
Right.
So you consider just the apical side of the Drosophila.
Yes.
But there are important changes in the third dimension as well that are
Yes.
happening during gastrulation, the cell becoming elongated, etc. So don't you think that to analyze, I mean, how is the third dimension involved in the analysis or important in the analysis of jamming and jamming? And the second question is about this liquid to gas transition that you show very nicely. And my question is, how different is that to EMT? Because some of the parameters that you describe are happening during this transition are very similar to some of the things that are happening during EMT. I mean, like the cell becomes softer and the nucleus becomes bigger.
Yeah. Two good questions. Thank you. First one about 3D. What about 3D? The answer is I wish I knew, and we have the same question in our airway epithelial layers, because those are pseudostratified layers, and we look only at the apical layer where there are apical rings, and when you go deeper, it's much more complicated. There are basal cells, and we think most of the propulsion comes from the basal cells, and the simple answer is we don't know. It's stunning to me that this works so well at the apical surface. And maybe it's because there are apical rings there that have certain mechanics that kind of conform to this. And I think that it's a terrific question. And I think the same thing applies in the Drosophila. And the answer is we just don't know. But the fact that we got such a simple answer looking at the apex, I don't actually know how to relate that into the third dimension. I think that requires someone to look at more deeply. And I'm not even sure how to go about doing that, but I think you put your finger on a really, really good question. Yeah, now about the cell scattering from the tumor and gas-like behavior. Yeah, I'm not saying that EMT doesn't happen. And in fact, we know that even in the advancing front, cells are going through what looks like a partial EMT. And so we think that could be coexistence of EMT and unjamming. We know for sure that at least in human epithelial cells, unjamming can happen without EMT. We know that for sure. What we don't know is whether EMT can happen without unjamming. And we suspect that when you get EMT, in fact, the paper that's on the bioRxiv suggests that early in EMT, you have to get unjamming as well. And so, but the really, again, you put your finger on a question that we're pondering. How do EMT and UJ, how do they interact with one another? Does one depend on the other? Do they interact? Are they independent? We think they're not independent in some cases, but we just don't know. And so I think those are two of the big open questions. So thank you for pointing that out. I think, and the short answer, I don't know for either of them. Thanks.
Thank you.
Okay, we've got a question from Dharma Pali. Does unjamming transition affect cell proliferation?
We haven't paid a whole lot of attention to proliferation in our systems. And I know that Javi Tripat in some of his systems, we found that yes, there is proliferation, but it didn't seem to affect very much the actual spreading in these systems. I think the short answer is I don't know. We haven't looked at that. So I don't know. I think Jinnah Park may know better than me. I think she might have looked at that a bit, but I don't know. Is the short answer.
Question from Leon de Boer. Do you think that intracellular pressure and/or molecular crowding plays a role in regulating the jamming, unjamming? Could
you say that one again?
Yeah. Do you think intracellular pressure and/or molecular crowding plays a role in regulating this unjamming and jamming?
That's an interesting question. And we've been thinking about something like that. When you look at tumor cells in tumors, and this is known to be true in ex vivo in tumors, and it's certainly true in Ming Guo's model. Cells in the center of the tumor, they are exposed to a higher pressure. That's just a fact. They have a higher pressure and they're small. The nuclei are smaller and the cells are smaller. The cells at the periphery are bigger and the nuclei are bigger. It's as if they're compressed. And actually, Minguo published a paper looking at water fluxes between cells, and it looks like the cells, in a sense, are compressible. In the sense, of course, they're really incompressible, but you can get water fluxes into and out of the cells that change the dynamics. And we know for sure also that when there's more molecular crowding in cytoplasm, the cytoplasm gets stiffer and it goes through a glass transition. So that probably plays a big role in unjamming as well, in terms of multicellular jamming. If the cytoplasm jams, which it can, probably the cells get stiffer and the cells will jam as well. But there is a lot of work we published on that like 10 or 15 years ago, looking at molecular crowding, and definitely can dramatically slow rate processes inside the cell. There's a whole literature on that.
Thanks.
I hope that answers your
question from Santosh Puyal. Is it known what triggers the switch between states? Is it the mechanical feedback from extracellular environment, or is it something coming from within cells?
That kind of gets at causality, and I don't know. Does the migration cause the unjamming? I mean, does the migration cause the unjamming, or does the unjamming permit the migration? I haven't been able to figure out an experiment that can unravel those. So that's a really, really tough question about causality. And if you have any ideas, if anyone has any ideas of how to unravel that, about which is the chicken and which is the egg, I'd like to hear it. Yeah.
We got a question from Mattia Serra. Mattia, thanks.
Yeah, so that was a very, very nice talk. I was wondering that with the shape variation or the jamming metric that you have, you want to assign the whole embryo or your whole in vitro system to a scalar field. So you sort of lose all the heterogeneity in space. For instance, in the Drosophila case that you showed, you focused on the ventral furrow, and then you wanted to have an overall scalar quantity that was assessing the state of your system. But in the whole embryo, there are many structures and heterogeneity. Right. So you can be jammed in some region or unjammed in some other region. Is that something of interest, or that you want?
For sure. I mean, because when you look at the whole gastrulation event, we were just looking at the early one, you see all kinds of interesting things. Mm-hmm. And there's probably are regional differences. Right where the action is in the ventral furrow formation is where the flow is, mm-hmm. and the migration is. Probably if you look at the basolateral side, you're going to see something different. So I think for sure, and this might even come into play in pattern formation in development. I think that in the paper by Sperlin et al. In development, that's in Celeste Nelson's lab, I think she found regionalized unjamming where the airways branch. So I think, for sure, for sure. Well, for sure, that's too strong a statement. I think so. Yeah.
Okay, thank you.
Okay, we've got a question from Elisa Jandri. Do more aggressive tumors have unjamming transition differences compared to less aggressive tumors?
Yes. Yeah, well, we know, in the ones we looked at. So we looked at MCF10A versus the MDB. And yes, but even though they were very different, they all seem to be able to be organized on this jamming phase diagram. Now, the jamming phase diagram is tremendously oversimplified. There are lots and lots of other factors that are not taken into account in that model, like migration along fibers and metalloproteases, and none of that is in there, but at least it shows the potential for a unifying idea for how you can explain different kinds of cell types. So in that simplified model, it would be, we think it's the propulsion, which makes the major difference. Differences in propulsion from more aggressive to less aggressive. That may be related also to EMT, to get back to Roberto's question. So it'd be nice if, everyone's always kind of looking for a universal theory of everything, and it might turn out that it's really much too granular, and what I've just discussed is maybe way too oversimplified. But on the other hand, there seems to be some logic that kind of explains, in one diagram, we can explain both the MCF10A and the MDB more aggressive cells. So maybe. Also, I should point out that Jae-Han Kim in my lab published a paper in BBRC a few months ago, where he wasn't looking at 3D tumors. He was looking at cell migration in 2D, and he looked at a whole panel of cancer cells of various degrees of invasive potential, and found that in 2D as well, there seemed to be an organizing principle that had to do with jamming, and it had to do mostly with cooperativity. The more aggressive tumors made cooperative swirls and packs that made the overall migration much greater. So it was all about cooperativity. So that's, again, it's not in 3D, it's in 2D, but it was very suggestive that cooperativity of the motion, which is typical of jam systems, might have a lot to do with it. Well, you're muted.
Sorry, I was muted. Is it okay if we ask a few more questions? I know we're running a little bit over.
Sure.
Great. So, I mean, the chat is completely packed with questions for you. So we got Leonor Menezes. Have you thought of testing other components of the extracellular matrix in your organoid substrate that have been implicated in cancer cell migration, e.g. fibronectin and laminin?
The answer is no. No, we haven't gotten around to that, and we're trying to focus more on the jamming dynamics than on matrix. That's a terrific question. Jacopo Ferruzzi who is one of the authors on that paper. He is totally into matrix, and maybe that's going to be in his future. I mean, somebody's got to do it, but the answer is no. We haven't done it yet.
Yeah. Question from Rob Tetley. In the MDA-MB231 spheroids, do the cells have epithelial characteristics, or is what you observe more of an EMT than unjamming transition, or is there a continuum between EMT and unjamming transition?
I think I don't know if it's a continuum, but I think they both must be operative, and you're right, and we think that those cells could be jammed not because of the nature of the cell-cell adhesion, but rather because of the crowding, and that would be more like the system that Oche Campas did in the zebrafish, where the cells are jammed not because of cell-cell adhesion or E-cadherin, but just because of either other adhesion molecules or probably just because of cellular crowding. So yeah, I wouldn't rule out EMT at all. Those are kind of EMT-like cells. So yeah, I would agree with that.
Question from Yolanda. Have you identified or studying the molecular switches that control or regulate unjamming transition? I think you kind of touched upon that already.
Yes. It turns out I'm the wrong person to answer that question, but Jinnah Park and Scott Weiss have published a paper where they did RNA-seq to try to look at some of the core modules that are associated with at least compression-induced unjamming in human bronchial epithelial cells. Turns out it's a really interesting story. It's published in, I think, Scientific Reports, I think. But if you look Scott Weiss and Jinnah Park, you can find that. And all that RNA-seq data is there, and we're continuing to pursue that. So yes, it may be that there's no one molecule or even cluster. It could be in different cells in different situations, you get totally different answers. But at least in compression-induced unjamming in HBECs, yeah, that's published.
Question from Shruti. The data shown here is for a spheroid of one cell type. Could you please tell us what would be the jamming unjamming transition in the case where there is multiple cell types, like in the case of a tumor?
Oh yeah, we haven't done that. It certainly is interesting to look at heterogeneity, and I can't remember. I just saw a paper about this somewhere. The question is, like, if you had cells that are mostly quiescent and you just have a few agitators in there, how much does it take to unjam the whole system? That's a really, really interesting question. I know people are starting to look at that, and we had wanted to do that, but just we haven't gotten around to that yet. It's a terrific question, because most things, I mean, a hallmark of real human tumors is heterogeneity. They're not all one cell type. There are multiple cell types. And of course, heterogeneity of cell types in cancer is kind of where it's at. People trying to unravel that and trying to understand evolution of tumor dynamics. I think what the unjamming does is it puts on top of that an overlay of mechanics. So not only are the cell types heterogeneous, but the mechanics is heterogeneous. And then furthermore, it leads to the possibility of mechanogenomics. The genes are going to express what gets expressed in the cells, but the cells are going to feed back and change the gene expression. So it makes the whole thing really, really interesting.
Thanks. We'll take just a couple more maybe because we're running at 10 past now. Chris Weston asked, how different is unjamming in endothelial cells and fibroblasts, brackets, if that does happen?
In endothelial cells.
Endothelial and fibroblasts.
Yeah, I don't know about fibroblasts. I think we have looked at endothelial cells a little bit. It wasn't so much in the context of unjamming, but it looks like it would probably, we haven't tested it, it looks like it would be the same story, but when you look at endothelial cells, usually you want to do it under shear flow, and under shear flow, they become elongated along the direction of the flow, and that gives you a different static baseline. So whether that makes things easier to unjam or less easy, I would think probably less easy because of the pneumatic order in those cells. But it's that ought to be explored, and we have not done that yet.
Thanks. Okay, just one or two more left. Yeah, there's so many. If you want to check the chat afterwards, Jeff, you can ask.
I will.
After.
I will.
Cool. So we got a question. I think Jen's trying to answer some questions on your behalf in the chat.
Oh, good. She'll do a better job than I would.
And then Jinnah Park is trying to answer lots of questions in the YouTube live. So thanks for that, guys. So question from Anna Huttenlocker. Thank you for the great talk. In asthma, there is a loss of barrier function. Is the unjamming necessarily without a loss of barrier function?
Well, in the air-liquid interface culture, Jen did this. Again, this is in the bioRxiv paper that's online. She showed that when you promote unjamming, that the barrier function does not degrade at all. It's stable. What in vivo, we don't know, of course, but you can definitely get a highly migratory, unjammed layer with no compromise of barrier function. And that's very different than EMT. So you look at that bioRxiv paper. That's where the data are.
Okay, we'll do the last question here from Badri Ekboti. Sorry if I'm pronouncing your name wrong. Do you think 2D migration or cell shape, do you think 2D migration or cell shape influence the 3D cancer cell invasion?
I'm not quite sure what that question means. If you can, it means can you predict from 2D assays what happens in 3D cell migration?
I'm not sure. Yeah.
If that's what it means, I think you can get a hint, but I wouldn't bet on it in any given cases. And we did find, again, in this paper by Jae-Han Kim, we did find that cells fell along a jamming diagram in that is to say, they show shape factors that you would expect, but they did not align themselves according to invasive potential. So there's more there than just the shape factor. So you can't predict from 2D necessarily what happens in 3D.
Great.
Yeah.
Thank you very much, Jeff. Yeah, sorry we couldn't get to everyone's questions. There's probably about 20 or more questions still in the chat there.
Okay.
So I think we'll finish it there. So for everyone that's watching, I think that's about 500 people who were watching at one point in total. Thanks for joining us. So next week, the same time, will be Robert Insel from University of Glasgow. All that information will be sent in the email as usual, which we'll probably send probably tomorrow. Please check your spam and junk folders because it may end up there. And that's all. So thank you very much for joining us, and we'll see you all next week.
Thanks for organizing.
Bye. Thank you, Jeff. Jen, is there anything you want to say, or is that everything?
Nope, this is great. Thanks everybody. And if you have more questions and you want to continue the conversation, we're on Twitter and we're happy to keep discussing this fascinating topic. Thank you so much, Jeff. That was a great talk.
Great.
Thank you, Jeff. Oh, and the one thing, if anyone is still listening, this has been broadcast live to YouTube. So we are going to put it up on the Cell Migration Seminars YouTube account. And so it will be available for the next week if you know anyone that couldn't attend and wants to watch. Cool. Okay. Thanks, everyone.
Bye.
Bye.
Thank you.
Thank you, Jeff.